Welcome to the Organic Wine Podcast. Matt Clark is an assistant professor of horticulture and enology at the University of Minnesota. He heads up the U of M's participation in Vitis Gen 2, a multi-institutional grape breeding collaboration that has a goal to breed new resistant grape varieties that taste delicious and can be grown with far less pesticide use than standard European grapes. Matt is at the cutting edge of the academic efforts that may result in the next truly great American wine from actual American grapes. The relevance of the ideas that Matt addresses in this interview cannot be overstated. The current wine industry around the world is built on a handful of varieties of the same species of grape. These varieties have not changed for hundreds of years, while the viruses, fungi, and insects that prey upon those grapevines have continued to change, evolve, and adapt. Someday, and that day is rapidly approaching, there will be a reckoning. New virus and fungal pressures will eventually overcome our ability to successfully grow these outdated varieties without massive amounts of crutches, chemical or otherwise. The main hurdle to allowing grapes, and therefore wine, to adapt is consumer perception, created by pervasively marketed ideas of the supremacy of European varieties. In this conversation, we talk about what Matt and other scientists are doing to create the future of wine. And I hope that it will help open your eyes to the immense potential and many options that are becoming available for grape growers and wine lovers if we just expand our palates and begin to embrace the change that is necessary to advance wine. I hope you find this conversation as inspirational as I did. Enjoy. The sponsor for today's episode is Centralis Wine. That's Centralas Wine. You can learn more about Centralis at centraliswine.com, and full disclosure, Centralis is my winery. I started Centralis because I noticed a disconnect between the values that many wine drinkers have and the kinds of wine they choose to drink. I wanted to give those of you who love wine an option to buy wine that reflects your values. So Centralis is built on two pillars. The first is that Centralis wine will always be made with, at minimum, organically grown grapes. And the second is that we will always tell you every ingredient that was added during winemaking. Our first vintage will be released very soon. In fact, it may be available by the time you hear this. And it's pretty limited, so if you want to get some, please go to our website, centraliswine.com, and sign up for our wine list. Or go ahead and buy wine. That's Centralas wine.com. We're also on Instagram at Centralis Wine, and I can't wait to share our wines with you. Matt, thanks so much for doing this. I actually am really excited to talk to you for several reasons, but why don't we start by, could you introduce yourself briefly and talk about what you do at the University of Minnesota, what your
Sure.
position is?
Yeah, my name is Matt Clark. I'm an assistant professor in the Department of Horticultural Science, and my position is sort of unique. My research area is studying grape breeding, grape genetics, and enology, which is wine science. And so in addition to research, I do some teaching and extension. That is translating some of the work that we do in research out to growers and winemakers across the state and the region. So I've been part of this project for just over 5 years as the faculty member in charge of running the grape breeding program. And our grape breeding program has been in existence since about 1908 or so. And then more
Oh,
recently in the 1970s, started focusing on wine grapes as we saw the development of a American wine industry. Minnesota wanted to be part of that. And so we focused to wine grapes in the last 40 years or so.
great. So you are one of the people at the heart of grape breeding in the United States, basically. There's University of Minnesota where you are, there's Cornell, UC Davis, those are the big three, and you're at the heart of that, right?
One of those people, yes. The other place that's doing some, not some, has been really important in grape breeding has been at the USDA. In particular, the researchers at in California at Partlier, and they focus a lot on table grapes, and have had really a big impact on that industry um, for a long time.
Oh, great. And I was going to save this question for a little later on, but since you brought up the USDA, at this point in history, what's it like working with them? I mean, from your perspective, are they helping to move things in the right direction?
Yeah, the folks at the USDA that I work with are primarily at Geneva, New York, and also then at Parlier in California, and they are fantastic collaborators. They have access to wonderful resources like the germplasm collections in California and in New York that encompass a lot of the plant materials that are the grape plant materials that are native to the United States. And so they have access to that material. Plus, they have a long history of doing research on grape. They're great collaborators in many different aspects of the work that we do, including disease resistance, using images to analyze, to extract data from digital images and things like cluster shape of grapes. We share pollen with one another. They're fantastic collaborators and great scientists to work with.
Oh, fantastic. That is really encouraging to hear. I love that. Um, do you ever think of yourself as like a monk, one of the like Benedictine or Cistercian monks of Burgundy minus the celibacy?
um, that's a great question. Actually, my undergraduate career was at a Benedictine university, so,
Oh, wow.
so I
I
know quite
mean,
a few of those monks and sisters. It's fun to be part of a tradition, and I think that's one of the aspects of this job is that what we do today, we may not see the impact for a whole nother generation. It takes about 20 years to develop a new variety, and so to be part of the process is really exciting and exhilarating, especially when you find something new or rediscover something old. Knowing that I'm just sort of one link in the chain of people who've been working on this project, and and for me, my team changes pretty regularly with changing in graduate students, undergraduate support, interns, and so forth. So we're constantly evolving with new thoughts and ideas and people, and that's the way it's always been, at least in the academic side of doing this type of work.
yeah, I love that. Actually, that was a question I was going to get to. I noticed as I was thinking of the things that I wanted to ask you about that I started phrasing my questions as we, like the grape varieties that we're developing, and I was like, I have nothing to do with this, that's all him. But I guess there's some, maybe it's just me, but I feel like when you're at a university doing the kind of work that you're doing, there is a sense that you are doing it for the public benefit as part of a bigger thing that is a, how do I put it, it's I don't know. It just leads people to think of it as shared by the public, and it's our accomplishments rather than just yours. And it sounds like you approach that from a very nice psychological perspective, which is you're part of something bigger and you're literally building the future for generations to come. I mean, does it ever come back to feeling underappreciated or unrecognized for your personal contributions? Or,
It hasn't yet.
okay.
There's a lot of tedious things that have to be done. There's a lot of grant writing and paperwork and all these other things that are kind of less fun. Um, but those things are also exhilarating. When you get the grant that you applied for, that means that you can hire a new graduate student or move a project forward in a new direction. That's really exciting. Although the workload of preparing to do that and the anticipation is exhausting, the rewards are great. And so that's one thing that I find really exciting about my position working at a university is I get to do all these different things and to take a new idea, find funding for it, and then run with it. It's certainly different than I think my colleagues who work in industries who are developing new products through breeding, whether that's corn or grapes or an ornamental plant. Um, we have a different bottom line that we have to meet compared to what they're doing, and so that maybe allows me a little bit more flexibility than some of those other individuals.
Got it. um this is uh, I'm going to just jump into a juicy topic. You just mentioned corn and breeding corn. To what extent are you making GMOs with with the grapes that you're breeding? I mean, it technically is a GMO, isn't it? Or is it not?
Yeah, that's a that's a great debate, a bit of a semantic one. So I think when we go down this road, it's important to define what GMO means or means to us. You know, plant breeders and humans for thousands of years have been selecting plants and making changes to them over a very slow pace of time. Grape breeding in the modern era means, choosing the parents and then choosing the seedlings that we like and moving those forward. So it's still a pretty slow process. And when people in general think about GMOs, I think what they're talking about is making edits um, through gene editing techniques or through um, agrobacterium mediated gene transformation to introduce genes or change genes. And that's not what we do in our program. Part of the reason is that there's very low consumer acceptance, it seems, for those products. There's a lot of
Mhm.
regulatory um, barriers to doing that type of work. And it's not clear if consumers don't want those products or if growers don't want those products. It's hard to focus on that area as a researcher. Um,
Got it.
so our new varieties that are being produced, at least at the moment, at my institution and at the others, use traditional breeding techniques. So we're not doing gene editing or GMOs.
And
That I should just comment that many of us do that type of work, but it's not necessarily part of our pipeline for a new variety. It's for testing gene function. It's for advancing the science, with anticipation, excuse me, that maybe someday those are products that people will want to grow or to consume.
right. Do you, and that would be using CRISPR technology, I take it, when you're doing the gene editing?
Right. Yeah. So I had a collaboration over the past year and a half, 2 years, where I worked with a colleague at the University of Minnesota who does a lot of work in that area to develop the techniques that could work in grapevine. And so we were pretty successful in showing that um, we could do that process faster and more efficiently than previous methods, although we don't really have any gene targets in mind at the moment to advance on that project. But like I said, we're developing the tools and the techniques. So if we do identify something that would improve um, how growers produce those plants or improve qualities for consumers, we'll be ready to go.
Do do GMOs bother you? Does that kind of editing bother you personally? Do you think there's a moral imperative that we shouldn't do it, or any other imperative that we should not do it, or are you fine with it, knowing the science side? And I don't know, how do you feel about
Yeah, I'm pretty fine with that. I'm not sure
it?
what your audience thinks about it. Just in terms of how biology works, it doesn't frighten me. My thought process on this has really changed over about 20 years, and I think that's logical and natural as we've learned more about the impacts. So I
Oh,
think, over 20 years since we've learned more about genomes, we've learned about gene editing and techniques, we've learned about the impacts on human health and the environment. Um, all signs to me say that these techniques work, that they can be done very safely, and with CRISPR-Cas9 constructs, they can be even more targeted, so that we can have specific outcomes. So I think that as a science, I think it's worth continuing exploring as one option for improving our food supply and food security. I think it's worth continuing to explore and to help people understand even better how those techniques work and can possibly improve quality of life on this planet.
yeah. I have a sense that the aversion to GMOs uh, probably arises from the fact that they developed hand in hand with things like Roundup and glyphosate, where they were modified specifically to be able to be grown in the presence of these pesticides and herbicides that would kill other plants. And I mean, that's, I mean, if I have any feelings about it, it's more about that, the fact that it arose, a lot of the corn and wheat GMOs that we have arose because we wanted to just broadcast pesticides and herbicides.
I think
And I
that's I think that's really true, and I would just add on that we've had a huge change in the agricultural landscape over the last 40 plus years, which includes big agribusiness coming in. We've
right.
certainly seen the decrease in the number of small family farms and conglomeration of large industrial scale farms. And all those things sort of went hand in hand. And as part of that were the development of things like Roundup Ready soybeans. And and I think it got mismangled, or excuse me, mismanaged, and the message mangled kind of about what was going on. And so I think it's this huge swirling vortex of fear and who controls the food system, who grows the food, how do we protect the environment. And a lot of people are disconnected from where their food comes from and what farmers actually do.
Yeah, that's the whole purpose of this podcast actually is to help reconnect people. That's my mission. So I'm glad you brought that up. And I wanted to talk to you for several reasons, as I mentioned. This is a really exciting thing because I'm kind of a geek when it comes to these things, and I'm just fascinated by grape breeding and the potential that we still have. Maybe this is the wrong goal, and I'll ask you about this, but to find a truly great American wine from a truly great American grape rather than imported varieties. Also, I think I want consumers to know that there's much more to wine than just Cabernet and Chardonnay, which I think you are on the cutting edge of some of those options. And also because I want wine growers to know that there are options besides spraying a lot of pesticides on a vineyard. And those options come from breeding stronger vines. So let's jump back, and can you talk about what the goals of grape breeding in your program are? And your program is called the Vitis Gen program. You're at University of Minnesota, but it's a multi-institutional collaboration,
Yeah,
right?
yeah, I can talk about a couple of different things. Um, where to start? So we call it Vitis Gen.
Vitis Gen, got
In
it. Sorry.
Latin, it's probably more appropriately to call it Vitis Gen, but we call it Vitis Gen. Um,
Go for it.
and in that program is sponsored by USDA NIFA, Especially Crop Research Initiative, which is a bunch of kind of mumble jumbo grant language. And we're in our second iteration of that project. And like you said, it's a multi-institutional research project that has a number of different research foci, including improving tools and techniques for genetics and genomics in grapevine, to advance breeding lines that have disease resistance, um, to educate people about the type of work that we're doing through extension and outreach. And so that project includes folks from all over the country. And then we also have an advisory committee that includes folks from industry, from academic sectors, both within the United States and around the world. And I don't want to go into too many details about that, but one of the key objectives that we're working on is developing new varieties with powdery mildew resistance, as
Mhm.
that's the number one fungal type pest in the world where grapes are grown. And with
I
some
can I
objectives,
can I can personally attest to that. My front yard vineyard. Yep.
yeah, so it's problematic everywhere grapes are grown, and so our project is really looking to understand the genes involved, to use DNA testing to help plant breeders like myself identify the parents that have resistance and then their offspring very early on, say in the greenhouse stage, when those plants are just a couple of inches tall, the ones that we know are going to perform in the field better. So one thing that your listeners might not know is that grapevine is native to the United States. There are 30 different species of grapes in North America, more or less, depends on how they get organized by the taxonomist, and then about 30 more in Asia. So the
So,
grape
and
that
you're talking species, not varieties, not like Cabernet
right,
versus Chardonnay, which is all part of the same species. You're talking about separate whole things.
you're exactly right. So the grapes that most people are familiar with all belong to the same species that were evolved in maybe the Caucasus region, moved with humans into Europe, and then really found its stronghold there in Europe, where it developed wine and table grape industries. And those are all the same species technically. So the work that we're doing is looking to these species in North America, as well as Asia, to find those that evolved with these fungal pests and insect pests on this continent, um, where we know that we have more diversity, and where those, because those plants evolved alongside those pests, we're anticipating and finding resistance to those pests. So my project in particular has been looking at resistance to phylloxera, which is an insect pest, and we've
Hmm.
identified some candidate resistance genes to the foliar form of that pest. Um, your audience
Yeah,
might
because we
might
know of phylloxera as a root louse, or transmitted by a root louse, which and that's why American rootstock, or North American rootstock, was used to as the rootstock for the European species of grape to resist that. But you're talking about a foliar, so
right. So
something that.
precisely, we're using that same sort of model in our breeding programs, not just for phylloxera, but these other pests, downy mildew, powdery mildew. Um, because these North American vines have those resistance traits in them, we just need to find those that have evolved to have those best resistances and then try to combine them into a new variety. So we're doing that through this Gen project. And then I would say that most of our grape breeding programs across this country are doing that anyway. But Gen has given us the tools to pool our resources and really make strong gains in that area. So not just the goal is not to just have a single resistance gene for powdery mildew, because we know that doesn't last over time. And that's not going to work for growers. It's specifically not going to work for any organic grower. We want to have multiple resistances into a single variety, so that as the pest populations evolve and change over time, and they will, and they do that very quickly, that if they overcome one of the resistances, there'll be kind of this, um, what's the best word? Several other resistance mechanisms in that plant that can respond and help to reduce that from happening. So we
And
call that durable resistance. So something that's
uh,
going to last for a long time.
do
We
you do
see
you do you behind closed doors call it CYA resistance?
CYA.
Cover your ass.
Oh no,
Okay.
no. And the reality is, when even I've been working in this kind of area for a while, we see that um, uh, plants like wheat, where they're working really, really hard to breed for resistance to things like stem rust, crown rust, that their resistances might only last 3 years. So they're fortunate, they're developing new varieties all the time, and they can because they get a crop every year. Grapevine, we have to take an even stronger approach, knowing that these varieties, we're hoping, last more than 15 years. Um, More than 20 years, and the varieties coming out of Europe, have been grown for centuries. Um, and we didn't know that they were susceptible to some of those pests until they came from North America into Europe. So we have a pool of resources, we call it germplasm, genetic resources here in the United States, that we're really turning to for these resistance mechanisms. The program at UC Davis has been working on and been successful in breeding rootstocks with resistance to things like phylloxera and nematodes, and more recently have released a few varieties with resistance to Pierce's disease, following the same sort of strategy.
Very cool. And I love this idea that I really, it really sort of solidified for me in just researching what you guys do and looking into the resources that are online about Vitus Gen 2 and the videos, which are excellent. I would turn anybody to onto that. Just some really great stuff about what you're doing. It's this idea that, you know, a few hundred years ago, we basically stopped breeding the cultivars that are now spread around the planet, and they haven't changed. They've just been clonally reproduced around the entire planet. And the evolution and adaptation of those, for the most part, has has stopped, has ceased. And yet the rest of the world, including the fungal pests and viral pests and insect pests, have all continued to adapt and evolve and get stronger. And at some point, there's going to be a reckoning, right? Like as has happened with other crops, where Cabernet just will not be able to be cultivated anywhere because everything will have caught up to it, and without massive crutches of chemicals or otherwise, I imagine. Am I painting a
Yeah,
picture that's kind of accurate?
yeah, I think you hit the nail on the head. And I would say that most other crops were doing this type of work and moving forward. And grapes seem to be the one that really like to hang on to these old varieties. A lot of it, I think, comes down to marketing, um,
Yeah,
and and that's been going on for 100 years.
yeah.
Even in Europe, where hybrid varieties were grown that had resistances 100 years ago, those those countries and the appellations and organizations within them, they didn't like these hybrid varieties for a number of reasons, primarily because it limited, in my opinion, limited their marketing abilities and capacity. And so
Right.
they, started calling these vines faux, fake, having problems, American
Mm,
hybrids, and that's really tainted the perception of new varieties as they've come out in this country, even though they're different. They may taste different, they look different, but so do the thousands of European varieties that are out there. They're also different and taste different and look different.
right.
But for some reason, we've got this baggage of kind of a smear campaign that started over 100 years ago on the varieties, even the new varieties that we're putting out today that in my opinion, can outcompete some of the European varieties.
Yeah, I've heard that marketing of French vinifera, for example, referred to as racist. If you look at it in context of almost any other kind of marketing, and I, just going back to what I was talking about earlier, this sort of dream of finding a great American grape to make a great American wine. I guess I just want to say it's not because I'm patriotic. I have patriotic feelings for the U.S. or against Europe or anything like that, but it makes sense to me that we want to let grapevines change and adapt and find something that works for the local climate and the local pests, and maybe even, I don't know, the idea of let's not make it about a variety, let's make it about a style or a region, so that within those frameworks you can continue to adapt and breed new things as the world changes, as climate changes, as pests change. Does that is that in embedded in what you're doing as well?
I I certainly feel a lot of those same things,
Okay.
and it's hard when you're up against these huge um, multinational companies that produce wines and try to sell
Yeah.
them, and their ideas about how consumers um, purchase, especially because they like to buy wines by name. They like
Yeah.
Merlot,
Yeah.
and all Merlots are going to be fine. Or they go through phases of their life where it's Chardonnay and spring and Pinot Gris in summer and Merlot in fall. I don't know. That's
Right.
I think
Rosé and
that's
yeah.
just the way that people really like to approach, or that I don't know if they like to. That's how they approach a lot of their wine consumption, and partly it's because, at least I think it's because there's so many options out there that it's by default easiest to go with something that you know.
And are you
Um,
seeing, I mean, I know you're not in the marketing and sales side of things, but have you noticed personally just a change in customer perception? I feel like the East Coast is a little bit better about this than the West Coast. Just because of the availability of hybrid grapes. Have you noticed a change in customer perception at all, or is it, you know,
the, yeah, I don't really know. I guess one thing that I can comment on in Minnesota alone is that we have nearly 80 farm wineries. Um, in early 2000s, I think it was like less than 15. And so we continue to see a growth in our industry and the
hmm,
number of grapes that are being grown and the wineries that are producing wines with these hybrid grapes. And most of their sales are at the winery. They aren't being introduced into restaurants. It's a hard scene to get into. If you go to any restaurant in the Twin Cities, you're going to find more or less the same 15 bottles of wine
yeah,
um, from the same supply chain.
yep,
So it's really hard to get their wines in there. and be successful. And liquor stores do carry some products. There's wine clubs and so forth. But I guess my point is we continue to see growth and people are visiting the wineries and buying their products. So to me, that says people are certainly willing to try them and go back and the experience and support the rural economies in Minnesota who are producing these products. So to me, that's exciting. We have a new variety that came out in 2017, and we're just starting to see those first wines this year. And so for me, that's like an exciting area where we have a new variety, a new opportunity to market a new product across the region and get people excited about something that's new and different. And it's a wine. I think hopefully people are willing to take that risk and try something. And interestingly, that wine's probably most like a vinifera than anything that we've released in the past. So our breeding targets are, despite the fact that we have access to different colors and flavors in the North American species that we use, our breeding target is to make something that's most like a vinifera. So,
Right. Yeah.
so it's a bit ironic in that way.
But I guess it's the path of least resistance at this point in history.
Yeah, and our earlier products, there we had a breeder in our region named Elmer Swenson, developed nearly 30 varieties, and they're very based in Vitis labrusca and Vitis riparia, and they have unique flavors and profiles, and maybe they don't always translate well into the wine, but they're different, and so people have tried these different wines for 40 plus years. And some people go back and try the new ones, and some people say, no, I just want to stick with what I know and what I like.
Well, I
Perhaps
know.
it's that Merlot.
Yeah, unfortunately, nothing against Merlot, but yeah, unfortunately, that being tied to old varieties. Marquette came out of Minnesota, right? And
It
Marquette's
did.
pretty big, right? It's been very successful. I know even organic growers on the East Coast are having success with it, and it does well. Even like in Vermont, you can grow it with.
Yeah, right. So Marquette is one of those. It's even being grown in Ontario,
Uh-huh.
which is kind of exciting. And that came out, I never get the date quite right, 2004, 2005. And it's a grandchild of Pinot Noir. It has some of those characteristics. It's cold hardy, maybe not quite as cold hardy as we once thought, but we've also had some pretty horrible winter across our region with polar
Right.
vortices over the last 7 years or so. We've had maybe three, and so
Yeah.
that's not something we thought we were going to have to deal with or we hadn't experienced in decades.
Right.
But yeah, Marquette is one of those. In my opinion, it's one of the best red wine hybrid grapes that's out there, and people are making some fantastic examples of those wines. And as you've mentioned, people are doing some organic production of that further east. And I think we have some examples of maybe not certified organic, but with organic principles even here in Minnesota.
Yeah, and how does that compare to the one that was developed in 2017? And does that one have a name?
Yeah, so that's called Itasca,
Itasca.
Itasca. Excuse me, and Itasca is a white wine.
Okay.
And what we really liked about that variety is that it's disease resistant. We still haven't identified which resistance genes. We have some ideas for powdery mildew. It seems to be very resistant, if not very tolerant, if not resistant to phylloxera on the leaves, which is important if that's something
Hmm.
you need to control for. It has a very low incidence of downy mildew. We are seeing a bit of black rot on that variety. But if you're controlling for in your vineyard for some of these other pests, black rot sort of gets taken care of in traditional practices as a kind of collateral. It just we take care of it if you're treating for powdery mildew, for example.
Right.
So we liked it for disease resistance. It was very cold hardy, even more cold hardy than its parent, which is Frontenac Gris. And then the third thing that's really critical is that our breeding program has really worked towards reducing the amount of acidity in the grapes. Something like Frontenac has about, on average, 16 grams per liter or 1.6%
Ooh.
total titratable acidity, which is very high.
Yep.
Its parent, the native Riparia grape, in general across our region can be even twice that, up to
Wow.
3%. So our
Yeah.
breeding program is working to reduce that. And Itasca comes in somewhere in the juice, I want to say between like 9 and 11 grams per liter, zero point 0.9
Oh,
to 1%. And then the
right.
wine, of course, it's even less.
Nice.
So that has moved the bar for acidity in our breeding program. La Crescent, another variety of ours, is pretty high. Marquette is still pretty high. And so what happens is winemakers have to do something to reduce that acidity. So they can ameliorate with adding water. They can use specific yeast to help drive down that acidity. Um, they can add sugar in the final product or make a sweeter wine to
Hmm.
counteract the acidity. And so those are all great tools for the winemaker, but for consumers who really want a dry style wine, it can be a limitation. So
Right.
Itasca has moved us one step closer in the direction of dry style wines, and we're really interested to see what winemakers actually do. And so we're just starting. I've just tasted my first commercial Itasca just this past weekend. And so we're excited to see there's, I want to say between 12 and 15 in Minnesota who have a product that they're offering right now from last year's harvest.
Are you did how'd you like it?
I did like it. I'm interested to talk to the winemaker to find out what they did and
Right.
how much they had to work with, because
Right.
at our research facility we don't have a ton. We probably have less than 40 vines. And
Okay.
so the research wines that we make are less than 5 gallons on average, and we're doing different techniques with them to figure out what we should be doing. And so it's
So,
a little bit different scenario when you're thinking about um, thousands of pounds of grapes and what the winemaker's doing. But I'm very excited to see where they go and what consumers think.
and so there's actual winemaking and wine drinking happening there at the lab.
We don't say drinking, so we don't get in trouble by the feds. We have a research,
Sampling?
yeah, sensory analysis and tasting is usually the words that we use.
There we go.
So we make a lot of wine that we end up discarding, and that's fine because as a breeding program, not everything is good, and so
Yeah.
you wouldn't want to drink those if you even could. But we do have a research winery um,
Great.
and and a license to do that. We don't pay taxes on that wine that we produce. That's why we don't get to drink it,
Right.
because the feds like to take their chunk of that, and I don't blame them. And so I have a enologist on staff, Drew Horton, who makes wines for the research program. We don't make anything if it's going to be less than half a liter. Um, we've tried smaller volumes, and they did that for a good 15 years or so. And we've decided to make larger volumes if we can, and if we can't, we're going to wait a year or two. For maybe we only have a single vine that we want to make wine from, and we'll harvest the grapes. And once we weigh them, we decide, what, let's just taste the juice and get some data from that.
Right.
But otherwise, for varieties that we're thinking about releasing or new varieties and even old varieties, we continue to do testing with different parameters. For example, looking at different harvest dates. We often tell growers wait as long as you can because the flavors improve. But that might not be true for a new variety. There might be some sort of critical limit where it hits some certain parameters, and that's the best time to harvest it. And the reason that we've often told people to wait longer is because the acids continue to fall, the flavors continue to develop, and sugar
Right.
accumulates. And those all generally sound like a good idea in a wine, but that might not be true for everything.
Right. True. Yeah, we're seeing that trend in California now for where we used to let things go very ripe, and now we're scaling back to on the flip side of that,
Right.
very
And
low
there,
alcohol,
you're
high
right, right.
acid.
Yeah, the limitation could be how much alcohol ends up in your bottle, and do you want to pay the taxes on that
Yeah, there's that too.
with that increase, and how do flavors change? So we work on that. We look at different yeast, different processing things. We're not a big enology lab, say like you'd find at Davis or Cornell, but our enology lab helps to support our breeding program, and answer a few questions along the way that we can tell growers, maybe this is, excuse me, wine producers, that maybe this is one way that you want to look at producing your wine, so they don't have to do this research testing on their own.
All the trial and error and the cost of that. Um, and as I understand it, now that you have something like Itasca with the DNA sequencing technology that you use, you don't have to crossbreed that in the traditional sense. Wait, develop new seeds, hybrid seeds, let them grow to the point where they can harvest grapes in 2 to 3 years and spend all that time waiting for it and then make the wine, taste the wine, and be like, oh no, this one didn't work. You can actually say there are these markers in the DNA, and we see that in this new seedling that we're growing, so we'll let it grow, and or we don't see that and we see these negative markers, so we can rip that out. Is that
Yeah,
true?
you're exactly right.
Okay.
And
So that's
Vitus,
sped up the process immensely, right? Less
yeah,
than a
and
year now for that cycle, I imagine.
yeah, so our VitusGen program has really worked to enable that in Grapevine, and I'm fortunate to have some really smart colleagues who know how to do that and advance that. And part of it is, finding the right investigators, postdocs, people who can really focus on that aspect and develop tools and techniques that work for many of us, as opposed to each of us doing one-off sort of projects to advance that technology. So we're very excited. We have a new marker system that we've put into place over the last 2 years, and that's been um, a big help because we can actually get genome-wide information, and included in that are the areas that we have the most information about. So powdery mildew resistance for one, seedlessness is another. The sex of the flower, you might not know, or your listeners might not know, that in the wild, grapevine is either male or female, and that the perfect hermaphroditic state is came out of domestication, and probably
Mm,
2 times. And so when we develop grapes, if you were to walk my vineyard, you'd find a lot of female plants, which is not great if you want to grow those commercially. You need to get pollen from somewhere. So
mm,
we have markers for some of those traits. And then myself and other colleagues are working on things like insect resistance, cold hardiness. Um, that list could go on and on. Any trait of interest that we're trying to figure out what a cluster shape looks like, the berry color. And so we're working to use, like you said, DNA tests or genetic markers to help inform what we're doing. Ideally, we would select those plants when they're in the greenhouse before we move them to a nursery or to the vineyard, because that's a labor step. They take up space, it takes money. And we've been able to do that for the last few years. Unfortunately, because of COVID-19, a lot of our labs got shut down right when we would be doing those processes. So this year, a lot of us have planted out into a nursery and then selected the leaf tissue. And so instead of those plants being just a couple of inches tall, my vineyard nursery that I looked at yesterday is about a meter for those plants. They grow pretty quickly. And so now we'll have to make our decisions. Hopefully, we get the data back that we need before the end of the growing season. And if not, we'll have to make those decisions later this winter about which plants get kept. So even though we've invested a lot of time and energy and money into developing the techniques, a global pandemic has changed how we do our work.
yeah.
But that's
Yours,
okay. It helps us understand kind of all the contingencies that we have to consider.
yeah. Matt, I could talk to you for hours, I think, and I could work with you for hours too. I love what you're doing. I know we want to keep this short, so I just want to say thank you so much for taking the time to do this and have this conversation. It's been super informative. I love learning more about this stuff, and I love knowing what you're doing, that these options are happening, and hopefully this will spread the word and help consumers know that as well. Is there any Could you give us the website or anything like that that you want to put out there for people to follow up with more resources and find out more?
Yeah, actually, you've mentioned the vitisgen2.org website, and I can just spell that real quick. It's VITISGEN, the number 2.org. And that's a great place to hear about some of the things that I talked about. Um, you can learn about the different grad students and researchers on the project. There's also I'm not finding it immediately, but there's also some links to what we call research in plain English. And my colleague Tim Martinson from Cornell Agritech, he likes to have us, when we publish a paper, write a very short description of what that paper actually means in plain English, so that our stakeholders and community members can know what we actually did and why it's important to them. And so those are housed also on this website. So it's kind of a great place to go learn about. Sorry, I'm at home. That's my dryer telling me it's time to go change it. Forgot about that. So anyway,
Time's up.
you can edit that out if you want. But if you go to vitusgen2.org, you can find sort of translations about the research that we've been doing, videos, as you've mentioned, articles in popular press. So we put out articles into some of the industry magazines. It's a great place to learn about what we're doing. So those are a couple. We had a story, I want to say 2 years ago, in Smithsonian Magazine. And if you just Google uh, developing the great North American grape, something similar to that, you'll find an article that outlines some of the work that we're doing.
Great. Yeah, I love that article. That was, I think there's a lot of great ideas brought up in that for
Yeah, I think
everyone.
it's really accessible to
Yeah.
a broad audience, and I think that's great. So,
Yeah.
that I think that gives you kind of an overview of some of the VitusGen things that we're doing. Each of the labs that participates is doing their own work, and then there's researchers, of course, that aren't involved in that project who really help what we do. I just, as a kind of a closing comment, University of Minnesota, we're not a big grape growing state. We're not a huge industry. We're about just over $80 million a year of economic impact. But when I look around my college and my university about how many people support the grape industry across the year, I added up over 35 different people at our university alone are helping the grape and wine industry in our state. And that really speaks to people's interest, to the impact that rural economies have in the state of Minnesota. And thinking about that, every state in our union has a winery, I think is really cool. And so it's something that I think we should continue to keep working on, advancing the science, advance our understanding about wines and why people like them, and continue to really develop people's lives and enrich them. And wine is just one way to do that.
Yeah, I mean, you're not just enriching the local Minnesota community, but I know the impact of your work is national. So thank you again. If you ever feel unrecognized, I recognize you and appreciate you.
Well, I
I think,
appreciate that. I just stand on the shoulders of everyone who comes before me, and the grad students and the technicians who do this work every day. I sometimes just feel like the paper pusher, but I appreciate your recognition, and I'd love to come talk more about some of the things that we're doing in the future.
that would be great. Yeah, thank you so much for your time now, and we'll definitely schedule that again. I would love to come back. I'm sure more questions will come up, and I think we're at the beginning of people tapping into the possibilities and potentials that spring from what you're doing, and so I think we'll see more and more interest in the future,
I hope
years to
so.
come. Yeah. Thank you so much, Matt. I really appreciate it. This has been a lot of fun for me, and I hope really enriching for everybody who listens. And I'll hope to talk to you again soon.
Sounds great.
All
Thanks,
right.
Adam.